Right, we were just talking about chlorophyll A and B, and that these ones will absorb these different colors of light. And then there's also carotenoids, and these absorb yellow and orange wavelengths. The name sounds like carrot because carrots have carotenoids in them. And so these ones are there for photoprotection, meaning photo light protection. They protect from bad light. They also broaden the color spectrum for photosynthesis, so they can actually help hand the energy over to these guys. That's some gross looking arrows. Okay, whoops. Back up. All right. We're still going over kind of background terms here. Next background idea is absorption spectrum. So this is going to determine the effectiveness of different wavelengths for photosynthesis. So if we take a look here, as white light passes through a prism, it'll separate out into these different colors. And then if we pass that through chlorophyll solution, what will happen is only certain wavelengths will pass through. So what they can do is pass something through. Now this has something called a galvanometer. Typically, if you do this in a lab, we use a spectrometer. But the general idea is that you take light and you pass it through the tube and you see how much of that light actually comes out the other side. So certain wavelengths have really high absorption. So if we shoot blue light at this, the blue light didn't come through. That's what this one is saying. Whereas if we shot green light through it, look, the green light went right through because the chlorophyll doesn't actually absorb that color. In the spectrometer, what you'll see is you'll get kind of, it's kind of like doing this, but a whole bunch of times all at the same time. All right, so typically if you do this, you will end up getting a bunch of mounds. And so if we take a look here at the absorption spectra, sorry guys, a little out of it today. Yeah, when I say mounds, here's what I mean. So we have the different types of chlorophyll. If we read here on the y-axis, it says absorption of light by chloroplast, and then the wavelength of the light itself. So here on these ones, you can see that these colors of light, they tend to get absorbed really well. These ones in the middle don't get absorbed well. These ones get absorbed pretty well also. Okay, so that's the absorption spectra. It's telling you what colors of light actually get absorbed. This is a slightly different thing, the action spectrum. This plots the rate of photosynthesis versus wavelength. These are a little bit different. So if you'll take a look here at the action spectrum, these are different wavelengths. So down over towards here, this would be the red area. You get with the plant's red light. Down here, this would be the purple and blue area. And then we've got green in the middle. So what this is saying is that if you give it light, here's how much photosynthesis it actually does. Back here on this one, this is saying, okay, if we give it light, this is how much of that light gets absorbed. So we have light getting absorbed here in the absorption spectrum. We have work being done with the light that was absorbed right here in the action spectrum. All right, so a scientist named Engelmann, he actually used bacteria to measure this photosynthesis rate in algae and came up with this idea of the action spectrum. And you can read more about this in your book. I'm not going to spend too much time on this. Okay, so remember when I said before to make a flow map? Well, this is probably a better area to start your flow map out with. So process of photosynthesis, here's where it would be a good idea to start that flow map and say, here's one thing leading to the next thing. Part 1, in the light reactions. So light reactions, electrons are going to get excited by the absorption of light. So here we have the energy of the electron in this diagram. A photon hits it and is going to donate that energy. It's going to get absorbed right here. That's going to excite an electron. And then what this is showing, this isn't saying that inside the plant there's like little electrons that literally jump up in the air. It's saying that's the amount of energy in there. So you see the light striking here, it's going to charge up that chlorophyll molecule. The electrons get really excited, and when they fall back down, they're actually going to release energy as heat and as light. Here's what this looks like inside the plant. We have this thing called a photosystem, big ugly purple blob, so you know it's a protein. And within this protein, we have these different chlorophyll molecules. This is all part of something called a light harvesting complex. So what happens is this photon comes and, oh, let me back up. So here's the plasma membrane. So there's one layer top, one layer on the bottom. This would be inside of a thylakoid. So here's our chloroplast inside an actual thylakoid right in here. Think about this as if you're looking at the cell, because remember that the cell membrane and the membrane of these chloroplasts is made of the same stuff. So now we're looking inside the membrane of a thylakoid inside a chloroplast, which would be inside a plant cell. That's where we're at. So the light comes in. It hits these pigments. These pigments are passing electrons to each other until they finally pass it to a special pair of chlorophyll A molecules. They pass on an excited electron to a primary electron acceptor, which is going to be a protein. All these different things that are doing this work in here, these are all proteins. When it says pigment molecules, these could be chlorophylls, they could be carotenoids, so it could be chlorophyll A, B, or carotenoids. Here's a diagram showing the actual structure of this thing called photosystem 2. So in here, these photosystems, the entire thing, like I said, is the photosystem. It's the collection of all of these proteins, and you'll hear them called photosystem 2 and photosystem 1. Ironically, photosystem 2 comes first. That's the first thing that gets hit with light. Why would they name it 2 instead of 1? It just so happened that they discovered this one second. They discovered the other one first. So kind of silly, but that's why they named them that way. All right. So light reactions, main idea. This again was more background. I know it seems like we're always going over background, but that's kind of how it is. So light reactions, main idea, we're going to use solar energy to generate ATP. So for reals this time, this is your first big idea within this unit, or within this section of the notes. It's using solar energy to make ATP and NADPH. Okay, now here is the actual part where you can actually make some meaningful boxes. Chlorophyll is going to get excited by light being absorbed. That energy excites an electron at the reaction center of photosystem 2. The electron is captured by a primary electron acceptor. You pause this as it goes, by the way. I mean, this is a lot of details here. So I'm going to kind of draw my flow map on the side. This excitation of the electron actually leads to water being split to replace that electron. And oxygen, this is the step, guys, where oxygen is formed. So this would be a good supporting detail here. So oxygen is being formed. Now this oxygen is actually going to leave the cell at this point through diffusion. Okay, so those were the first 3 steps. Chlorophyll gets excited, passes an electron, or passes that excitement, so to speak, the energy via electrons to photosystem 2 to the reaction center, and water gets split to replace the electron that was lost right here. 4th step, energy gets passed to photosystem 1 via the electron transport chain. We're familiar with this because of respiration. So the electron transport chain, again, that's a bunch of proteins that are good at handling electrons. In this 4th step, energy is used in that transfer to pump hydrogen to the thylakoid space. So here's our stack of thylakoids. These hydrogens that were inside them are getting passed outside and into the stroma. So here's the chloroplast. Chloroplast, again, sitting inside a plant cell. So it's getting passed to this part right, oops, I'm sorry. Nope, not right there. Just outside the thylakoid. Boom. Okay. And from here, chemiosmosis is going to produce ATP. So this is the same step that we saw happen in the mitochondria. Inside the mitochondria, this was happening in the folds of the cristae, so hydrogens got pumped into this inner membrane space, and then they came through this special turnstile ATP synthase in order to make ATP. Same thing is happening here, only here they pump the hydrogens out of the thylakoid, and now the hydrogen comes back through, and that's going to make a bunch of ATP. ATP. Okay, and we say that this is done by photophosphorylation. Photo for light, phosphorylation for taking the taking phosphate and adding it to ADP. So ADP gets turned into ATP because we've added a phosphate group to it. Okay, and this says step 5. It's really kind of a detail here within step 4. If I could redo this right now, I'd make that step 5, and I would make this a supporting detail. So switch those. Sorry here, guys. Alrighty, and then steps 6 and 7, in those an additional photon hits the next photosystem, and the energy from that is moved to actually do the same type of thing again. So the energy has come through, it's excited it, it's created a bunch of ATP, but the electron can still be moved around and used to do work. After this happens, NADP is going to get reduced to NADPH. So here's another diagram showing this in more of a visual way, lining it up step by step. So in step 1, light strikes these pigment molecules, and then these pigment molecules are going to pass electrons until they finally reach this primary acceptor. Again, the cell membrane would be like this. So we're inside the actual thylakoid. That's what this is looking at. We're looking at the membrane of a thylakoid right here. This is my magnifying glass. Whee! Oh boy, it's getting out of hand. And then that thylakoid would be in a stack called a grana, inside a chloroplast, inside a plant cell. Okay, so this is what we're looking at. And inside this membrane, this is getting excited. This is inside the thylakoid. Okay, from this is actually going to split the water because these electrons have to replace the one that was lost. And this is where oxygen is being produced. These electrons get passed down through an electron transport chain. The electrons end up here at another molecule, another chloroplast molecule. From there, another light photon excites these ones. You see it passing them around. And then that is used to boost up the energy, goes through another electron transport chain, and makes NADPH. So if that's not making any sense to you, and you need to just kind of recap it for yourself, just think, okay, so these thylakoid membranes here. So here's we're looking at the top of the thylakoid membrane, outside, inside. We're looking at this little piece right here. So big picture, this is outside, this is inside. So big picture, light is striking this once, that excites an electron. The electron is being passed through an electron transport chain to create ATP. That electron gets excited again and gets used to create NADPH. So this whole system has been used to create ATP and NADPH. Here's another diagram. I like this, another visual way of seeing this. So this photon mallet guy smacks this seesaw, excites the electron, gets passed through an electron transport chain to do work. The electron gets excited again by another photon mallet, and then this worker is going to use that to create NADPH. All right, and like we said before, photosystem 2 and photosystem 1, it goes in that order. Why? They discovered this one first, then they discovered this one. Okay, there's another process going on here called cyclic electron flow, and that uses only photosystem 1, and that produces ATP for the Calvin cycle. So it can use this. It doesn't create as much ATP, but basically if you get hit with light and it excites it, this could pass down and make ATP and then make more NADPH, or in this case, this could just make less by going through this process over and over again. So it's kind of like running your car. If you had an electric car or something, running it off of gasoline, you've got a lot of power here, or you could run it just off the battery, or you could use both. All right, great diagram. I would pause the video and make sure that you can actually explain what's going on here. So what this is saying is if we look in these spaces, same process is happening in the mitochondrion as this happens in the chloroplast, and that's that we are exciting electrons using some kind of energy. In the case of the mitochondrion, we got that energy from breaking apart food, and then we passed electrons across that paid to pump hydrogen into this inner membrane space. In the case of the chloroplast, the energy from light, that excited an electron, passed through the electron transport chain, paid to pump hydrogen across the membrane here. In both cases, hydrogens, protons are going to go through diffusion through ATP synthase, and this process right here where the hydrogens go through, it's also called chemiosmosis. That is used to phosphorylate ADP into ATP. So same process happens in both of these organelles. Just ultimately a different outcome later on. And diagram number 400, we've got the same thing going on. This one may be a little more helpful for picturing the exact steps going on, so I would encourage you to pause this and go through it. I'm not going to take the time because I've already kind of hammered that to death in the last couple of slides. But see if you can go through this and actually explain what's happening in these steps 1, 2, 3. All right, and that's where I'm going to leave off for this one, and then we'll pick up in the dark reactions in the next video.